Okay, 10:30. Let's get started. Taiwan, can you hear me?
Yes.
Okay, great. Let's get started. Good morning. So, first thing first, homework. For this year's homework, I asked you to write an essay. Okay, my favorite experiment in science. Okay, so write about your favorite experiment in science and email that as a PDF to my email address, which is here, right? To Wang at iSIMS, blah, blah, blah. In the title of your email, I want you to put exactly this prefix, GFLS 2019, underline, and then your name, right? This is going to help me search for the homework because I receive lots of emails and I'm going to search your emails by using this keyword, okay? So don't forget to put GF, Global Frontier Life Science, right? 2019 in the title of your email. I want PDF, not Word. Every time people send me Word files, some students will send me Word files, and I take points off because I ask specifically I want a PDF. No Word files, please. Okay. I don't like to receive your homework on any day. I ask you to turn them in during this week. So July the 8th to 12th, that week. Anytime during that week, send send me the PDF. Okay? But don't send, like even if you have already written a really beautiful essay, oh, I can't wait. I want all time to read it right now. But don't send it before the 8th, okay? 1 to 2 pages. That's the length. This is A4 size pages. And I want you to write how include these elements in your essay. So I give you an example. It's a very simple example. I made it up. Well, the experiment is real. I didn't make up the experiment, but I made up this essay. So you're supposed to include all these elements. First, who did this experiment? Okay? Right? Who did this experiment? Relative, when? Not like, not, it doesn't have to be on the same, on the, at the day resolution, but you can say, well, 500 years ago or, 10 years ago or something like that. And where were the experiments conducted? Um, and what exactly is this experiment? What did the author, what the person trying to find out? And how is the experiment designed or done? Okay. And finally, importantly, why is this experiment your favorite experiment? Okay. Question? So your grades will be calculated based on your attendance and also the grades on this report. For the people who came in late, I'm giving out homework assignment. Okay? And the homework assignment again is to write an essay, my favorite experiment in science. A lot of people write about the experiments in history, like, the experiments the physicists used to measure, to define how long a meter should be, for example, right? But some people also write like, oh, my favorite experiment is PCR because I do it every day. But that's not what I want, okay? Every year you say, oh, my favorite experiment is CRISPR because I use it to, in my experiment to knock out the genes, and that's not the reason I want. You can write about CRISPR, fine, that's fine, but because it's why it's favorite to you, you should be able to tell me why the CRISPR is so significant. It's so important, not to you, but to the whole humankind. Okay? Clear? Everything's clear. Okay. So again, for people who came in late, make sure in the email you put this GFLS 2019 in the title so I can search. Okay? Taiwan, do you also want to send me your homework or not? You can discuss later. Either way is
Yes.
fine with me. Oh,
Yes,
you
we
want me
will
to
send
send?
you
Okay.
the homeworks.
All right, so please follow the same format then. All right? Thanks a lot. So that's clear. Okay, then for the rest of the class, lucky you, you're just going to watch YouTube. Yeah, I'm not kidding. No, I'm kidding, but I'm not. Give me 1 second. Let me switch to YouTube. Okay. Oh, how do I do that? 1 second. I think I need to share screen with you so you can see what I see. All right. Okay, there you go. Let me turn off the light so you can see a bit better.
Excuse us, we cannot hear the video.
Yes, maybe it's really important for you to hear it.
Tina, tell us a little bit about Cody. Cody is blind and autistic. Wow. We found out
It's great now.
really early on. Listen, and eyes just went huge, and he started singing, and that's when I that's when
I like very much. Taiwan, why don't I email you a link and then you can. And
Yeah, we can hear now. You can continue play.
right, the only problem is that I don't have the audio from my computer connected to the what's that, the conference system.
Yeah, but we already turned the microphone louder so we can hear from
You
the
can
video.
hear some.
Yeah,
Okay.
that's right.
All right.
Okay.
Thank you. Sorry about that.
oh my gosh, entertainer. So through music and performing, he was able to again live hard. Actually has saved his life. Playing. think I probably kind of like abused art, passion, your voice. Through music and performing, he was able to stand, live in the world, art, to do actually has saved his life. Like, well, we'd love to hear you. Go for it. I'm ready.
I've been to so many places in my life and time. I saw a lot of songs and I made some bad ones. Acting out my life in stages, 10,000 people watching, yeah. We're alone now, and I'm singing the song to you. I love you in a place, oh, there's no space or time. I love you from my life. You're a friend of mine. When my life is over, remember we're together. We're alone now, and I'm singing the song to you.
Guess once in a while we can do a YouTube class, but not today. Okay. Um, what do you think? When you close your eyes, you hear Sam Smith? No? Oh, maybe his name is Cody. But yeah. So what you just saw is this individual. I mean, I don't know if you heard it well. Next time I should double check the audio. But it's really extraordinary, you know, and talented. But he's also disabled. He's blind, and he's autistic. Sorry, the spell. He's autistic. Okay. So the question that I try to um, talk a little bit to you is about the sort of like how learning and the memory helps us to learn about the world, the environment, and learn how to interact with the world in the appropriate way. Okay, so I don't know if you have the same experience, but when I was a kid, I had this question that why do I see a strawberry in red just like anybody else? You know, you think that the knowledge, this is when I was small, so I was thinking that all my knowledge or, all these things should be taught by my parents or by my teachers, by books, but nobody has told me that the strawberry is red. So why did I get this knowledge? Where did I get it? And what happens if I see blue? You know, you just saw individual Cody. He's very extraordinary. He's very unique. He's different, but he's very different from other people. So I had the question. Then, and then now I think I understand a bit better. We are not born to be able to see red strawberries. I'll give you a couple of more examples. This is a chessboard visual illusion. Anybody see A and B in the same color or different color? Different color? Same color? Same. You see the same color. Yeah. Somebody see A and B have the same color. And different? And most of us see different. They're different. They are different. I mean, there's no yes or no. If we just connect A and B here, you will start to see them in, most of us will see them in same color here, right? So, in terms of color, oh, I should use the board so Taiwan can see. So in terms of the color intensity, spectrum A and B, they're the same, but our brain perceives them differently. And the reason for that is that first we have some knowledge about chess game. We know that white and black should come alternately. So we expect A to be black, and we expect B to be white, right? So, and then what is column doing here, right? If we just see that, oh, A and B, they're different, so different, this is also black. I said, oh, maybe the person who, made this chessboard made a mistake. But here, we have an object right here that is projecting shadow. So, oh, that makes sense because B is white, but it's in the shadow, so it becomes gray. So our brain makes sense of it and gives us the conclusion. So we say, oh, A is A and B, and and A is in the light, so it's brighter, right? So what I'm trying to tell you here is that the way our perceptual skills, the way we perceive the world, is based on our knowledge, is based on our experience. Okay. So again, oh, how do I erase this? Oh, perfect. Just there. Okay. All right. So again, we have the sensory input. Again, we have the sensory input coming, and our brain can, based on the sensory inputs, our brain will calculate the best actions. We can have action A, action B, action C. You get the same phone ring. If if it's your phone, you will pick it up. If it's your friend's phone, you won't pick it up, right? So you have choices of your action. Now, then we have experience. Oh, sorry about the animation. So now we have experience coming in. And the experience is what experience does to our brains to bias us toward one choice over the other. And to be able to do this, learning and the memory is important. Okay, so that's the research that I'm interested in, and I want to do it through my own research. So how do we do this? Before we go into the mechanisms of learning and the memory, I want to tell you a bit. I need to tell you a bit about the synapse. Anybody knows? Has heard the name synapse? Yeah. Oh, good. Good. I have quite a number of people who um, know this word, the synapse. So here's a picture of the synapse. We have a pre-synapse. We have a post-synapse. There's a small gap that's about 20 nanometers across. And when there's an electrical signal coming in from the pre-synapse, so from a neuron A, the neuron A will, the electrical signals will cause these little vesicles dock and release the neurotransmitter's contents into this small cleft. And on the presynaptic side of neuron B, there are these receptors that are regulated by these neurotransmitters. And when they open, there's going to be sodium current, mediated currents, calcium, potassium current that should sort of transmit the signal from neuron A to neuron B. All right? And the reason that I was able to tell that, oh, the information flow is like this, is that it's these synaptic vesicles. And in our brain, in the mammalian brain, we use glutamate, GABA, or glycine, acetylcholine, etc., etc. Many, many different transmitters, so that can be used to regulate the information flow in the brain. And for example, these two neurons here, right here, will be connected this way. In the human brain, we have about 100 trillion. So 100 trillion will be, let's say, billion 10 to the 9th, so trillion 10 to the 12th, so 100 trillion will be 10 to the 15th. Right? Very big number. A lot more than the GDP of Japan. Okay. So synapses are important for memory because of two properties of synapse, specificity and plasticity. So I will show you some examples right here. When you see things, right, you have these, receptors on your retinal cells. In your eye, you have these cells, and then these cells will make a synapse onto the cone bipolar neurons right here. Right? So here, let me get this. So you get one set of synapse right here. And these cone bipolar neurons then go into the ganglion here to make another set of synapses, right, to the ganglion cells. And then this information go into your central nervous system. So two synapses, the light information is in. Here you have another set of synapses, 1 and 2. And you can see that the synapses, they do not cross. For example, this cell will not make synapses to these on cells, and these neurons will not make synapse into the off cells. And also the synapses are organized in a way that they're architecturally specific. And it's because of this level of precision that we can actually have a relatively reliable information transmission. Okay, so next, plasticity. Plasticity, plastic? Plastic means you can change, right? Change, elastic. Here is the synapse between neuron A and the B. And here is the connection strength between the two neurons. Right? Yeah, we can measure that. We can look at, if we fire this neuron, we're going to get this much of a response in neuron B. But after we stimulate this neuron repetitively, many, many times, let's say 100 times in a second, 100 hertz, then we get a much bigger response now in neuron B. Much bigger, right? So here, this much bigger. So we say that, in this case, we say that the connection between neuron A and the neuron B is strengthened. Okay. Strengthened. And if the connection goes from this way, then we say, oh, the synapse is weakened. So, the synapse can be strengthened and weakened. And they can also be created and eliminated. So the number and the connection strength of synapses are plastic. For example, oh, so okay, let me help you locate the synapse in the brain using a mouse brain now. This is a C57 black 6 mouse. So you're going to see that in this mouse brain. Okay, so here then, this is actual estimation of the number of the synapses in the visual cortex. So, so first you look at the black line right here, right? This is birth and the childhood, adolescence, adulthood. You can see there's very quick increase of the number of the synapses in the visual cortex, and it peaks around at the border of between childhood and adolescence, right? 11 to 14 years old. And there's a gradual decrease and then stabilize and then further decrease, aging related decrease. Now, the purple line is following a patient in autism. So like in Cody that we just saw in the America's Got Talent 2019. You can see that Cody will have way more synapses, and it doesn't come down a lot during adolescence, and then it's stable. What happens with the patient with schizophrenia, so the green line, schizophrenia, we get this nice increase But then there's way more elimination than normal. So we ended up having very low number of synapses during the adulthood, and the number keeps stable. In the case of Alzheimer's, we have normal, everything is normal, normal, until in the late adulthood. Then we have a quick drop, we have sudden drop, and this happens before the neurons die. And once neurons are dead, they are not regenerated. Okay. So for me, this is very interesting because it tells me that the way that the synapses are made, the timing that the synapses are made is right coordinated with the birth. So we have this increase right here because a lot of the things we are experiencing around birth, after birth, I think it's going to be integrated into the synapse formation. So in a way, the synapse formation is experience dependent. And I think that's important for us to learn about the surrounding environment. So I want to understand the mechanism of this. So how do I do that? So because it's just a short class, I'm just going to cut the chase and tell you then, we know that the genome information is very important for us to learn the world. It's important for us to learn. So, and we know that for any type of persistent memory to be formed in the brain, we will need new protein synthesis in neurons. That is also known. Right? So, thank you. Okay. So I thought, well, how about RNA molecules? RNA molecules are the direct readout of any genomic response. And they are also the input molecules for protein synthesis that is required for any form of persistent memory we know on the Earth. So it must be a very nice of class of molecules to study. So then I got very obsessed about RNA molecules. I'm going to skip lots of things here. So where do I want to study about the RNA molecules? As I've just shown you that the synapses are very important for memory formation and also for cognitive function. So I wanted to study the molecules of these RNA molecules that come off here from the chromosome, and now they get recognized by the proteins. They form these large granules, and these granules get transported by the kinesin motor proteins on the microtubule into the very distal site of the neurons. So this has been known for the past 30 years. So okay, then it will be very interesting to study the RNA molecules right out of the synapses, and they can be translated, right, to make proteins right out of the synapses that it can be used by just that synapse to either grow or to, shrink, to switch on and off the connection in the brain circuit. Okay, so if you're wondering how do I select a research target or a research object, this is something that I can share with you. This is my experience of how to narrow down my question to a very defined location, the spines, the synapses, and the RNA molecules right here. Okay? So it doesn't matter what kind of question you want to ask in your scientific career. You need to further define it to the level that you can actually approach it to design experiment to specifically test it. Okay. How are we doing on time? So we have 10 more minutes, right? Okay, so how do I prove this? How do I look at whether during learning there's this synapse-specific protein synthesis Well, I go to a very simple model system, sea hare, aplygia. In aplygia, they only have, in this ganglion right here, they only have 24 sensory neurons and 6 motor neurons. So what we can do is we take one sensory neuron and then one motor neuron, right? Very simple, 2 neurons. Throw them into the dish, the culture dish, and they somehow can recognize each other and make synapses from the sensory neuron to the motor neuron. Then what we can do, I have to back up one point. So in this system, we can find that some RNAs are restricted to soma, but then some RNAs are localized in the very distal sites of the sensory neurons. Okay, this is great. So we know that there are RNAs that are localized to the synapses, and it's not every RNA, so it's selected. Okay, and we found a messenger RNA that actually encode a neuropeptide, small neuropeptides, that the RNA is highly, highly concentrated at the synapses between the sensory and the motor neuron. Then what we had to do is to find the elements in that mRNA, to find the elements in there, either the sequence element or some structural elements that can drive any RNA into the distal site. And we found those elements. We found elements that are in the untranslated regions that you don't have to remember this, but I'm just trying to use this as an example to tell you how to set up experiments. You need to find your elements. We found the elements that they can drive the RNA into the distal sites of neurons. So without those elements, you see there's no RNA in the distal site, but with those elements, now the RNAs are out. Now, with these elements, so what's next? Then we set up systems, okay? We set up a system that we have a sensory neuron right here, we have a motor neuron here, we have the synapses here. Then we put in a little pipette here that has serotonin in it, and also with a blue dye. We know that if we puff serotonin onto those synapses, we can induce the strengthening of the synapse. Just like to show you the strengthen goes up. We know that. And we know that when these animals are learning, the interneurons in these animals will release serotonin. So now, again, with all these elements, we provide a little pipette to puff on the synapses. We have a blue dye, and then we put up a pump to wash away the blue dye from the stimulated sites so that in the same neuron, only one set of the synapses will be stimulated, and this set of the synapses will not be stimulated. Okay? So this is an extra picture. We're going to ask whether the serotonin is going to stimulate protein synthesis in just a subset of synapses or not. So that was the experiment. And when we did that, well, there are other technical, designs I used. I'm not going to go into those details. When we did that, we found that although the RNAs are localized to both sites, only in those serotonin, so serotonin is abbreviated as a 5-HT, just you know, then only at the stimulated sites, we saw this increase of protein synthesis, while the unstimulated neighboring synapses remain the same. What this tells us is that a neuron can make thousands of synapses, but they can independently regulate the protein synthesis at individual synapses as a way to regulate them. And I guess the implication of this is that we can specifically strengthen or weaken synapses so that we can maximize the computational capacity of our brain, for example, for memory. All right? And that's a science paper. So now you know how easy it is to publish something in science. And EM electron microscopy has also shown that the polyribosomes, you can see these polyribosomes, right underneath the spine of this neuron, right? Again, supporting that, well, because polyribosomes are only assembled during active translation. So this really suggests that there is ongoing protein synthesis right here to supply, to give the local supply. Good. Now we know a lot more about the protein complexes. So these proteins are not only made at the synapses, now they have to form all these structures, the complexes in the presynapse, in the postsynapse, but also in the cleft between the pre and the post. There has been over 1,000 synapse proteins identified, and they are associated with over 130 human diseases. Psychiatric, neurological disease, childhood developmental disorders, and some of them follow the Mendelian genetic rule, and some of them don't. So there's more complex genetic mechanisms there. Okay, so 5 more minutes. Okay, so how do we further understand during learning what is going on with okay. So now we know there are a lot of mRNAs that are localized to the axons and dendrite synapses in the neurons. They are used in many, fundamental functions of neurons. And the dysfunction of this mechanism has been associated with many diseases, right? Huntington's, spinal cerebellar ataxia, ALS, Fragile X, autism. And I want to draw your attention to this one, the spinal muscular atrophy. And the reason that I want to talk to you about this specific disease is that just 2 weeks ago, US FDA approved the most expensive drug in history. 2 weeks ago. So this is the Zosyn SMA. Okay? So this drug targets that disease that I just talked to you about, the spinal muscular atrophy. Anybody guess how much this drug costs or this treatment costs? It's a one-time treatment. $1 million? No, I don't know. One time. It's one shot. One shot. $1 million? Too expensive. $500,000. I don't know. Somebody, I mean, come on. This most expensive drug ever in history. And it's targeting babies who are born with genetic deficits that results in their muscle atrophy. So most of them die before they turn 2 years old, right? They will stop breathing eventually. So how much would you pay? Let's say you have a child that is born with this disease. You see that your child stops like moving and she's strange. You know that if you don't do something, then your child will die before they turn 2. How much would you pay? 1億円。2億円. You have a choice now. I know most of you don't have children yet, but good to have a simulation. I'm not trying to discourage. It's very rare. It's very rare disease, so you don't have to worry about it. But just put yourself in the shoe of the parent. If your child is born with this genetic disease, and it takes one shot, just one treatment, and which has long-lasting effect on your child. It's going to save his life, her life. That's it. And this treatment is more than $2 million. One shot. Okay. But the trial, the effects was dramatic. And as a scientist, I worked closely with the people who are involved in development. And they asked me, how much do you think, what do you want to put for that? I said, wow, at least I would charge $5,000. And my friend said, "Oh, Chan, think as like a business. You're thinking like a researcher." Anyways, it's just so I was very off of in terms of the price. Yeah, 2.125 million US dollars for that one shot. So I don't think I have time to actually tell you what exactly we do. In my lab, we try to image, we try to see the RNA, to analyze the RNA, and also to mimic the RNA regulation using synthetic biology approach. So I can show you probably one movie of, I'll just show you one movie that is that came out of the study. So this is beta-actin mRNA in a living cell, in a living epiblast cell. You can see this little red dots. So these are the beta-actin RNAs. Yeah. So I hope what you, yeah, it doesn't come out very clear on the screen, but I hope you can see that these RNA molecules are very mobile. They're moving everywhere in the cells. So, it's kind of cool that our genetic information is not just restricted to the nucleus. Because of RNA molecules, our genetic information is not restricted to the DNA in the nucleus, but they can be delivered actually to the corners of the cells, to every corner of the cells, and the cells can use those genetic information. And that is just published in 2014. So not very, it's not a long time ago. So our genetic information is very mobile. And we don't know exactly what these movements mean, right? How do we analyze the meaning of movement of genetic information in the cell? It's a challenge, but we have some clues on how to do that. Oh, well. So here, for this research that I have done, I started from Dr. Akira Kudo's lab in Tokyo. This is Tokyo Tech, and moved to U.S. First started out in Birmingham, Alabama, to study the GABA transporters. So GABA is one of the neurotransmitters. So I was studying about the chemistry of GABA in the brain with Dr. Michael Quick, and then moved across U.S. to UCLA in Kelsey Martin's lab. In her lab, I directly started, probe the molecular mechanisms, learning and memory, RNA localization, and then moved back to a chemist lab, Okamoto Akimitsu's lab in Tokyo, to develop RNA probes so that we can visualize RNA molecules, and then came to Kyoto University, starting my own lab. So I'm Chinese, but you can see that all my scientific parents, mentors are Japanese or Americans. So it's been a quite interesting and dynamic scientific career for me. Okay. Anyways, so when we're doing this RNA imaging work, we started to realize that RNAs can be modified extensively because we were actually engineering those modifications to the RNA molecules. And that's quite different from what we have learned from the textbook. And this can be a huge gap. Let's think that there's an RNA that has 6 nucleotides, AACCU, just 6 nucleotides. And because each of them has you have 4 possibilities. You can have an A or a C or U or G in each of the positions. So you can have 4 to the power of 6 ways of coding using this RNA. Okay. Now, think about like a chemist. The chemist will tell you, well, if you put a methyl group right here, you're going to get an M6A. And if you put a methyl group here and another methyl group here, you're going to get M6AM. And then if you put a methyl group next to M6A right here, this is M1A, and it generates a positive charge. And you can do this to the cytidine, you can do this to another cytidine and uridine. And now, because all this chemical moiety also carry genetic information that can be recognized in the cells, instead of 4 to the power of 6 coding for 1,000 something, we actually, you can have to up to 171 plus 4 to the power of 6 ways of coding. So our array can actually has a lot more coding power than we previously estimated without the chemical modification. So I'm running out of time. So I'm going to wrap up just to tell you that this kind of RA modifications can change the channel properties in the brain, can change the signal transduction efficiency in the brain, and the abnormalities of these modifications have been associated with seizures, depression, schizophrenia, suicidal behaviors, etc. And when we started to probe whether that the local transcriptome is modified or not, when we started to probe the coding capacity of this local transcriptome, use the next generation sequencing techniques, which is empowered by the Human Genome Project, which was finished in 2002. So what this allows us to do is to align all our reads from the sequencer back to the reference sequence of this of our genome. In this case, it's the mouse genome, and then we are able to actually have a definition of the synaptic transcriptome and the epitranscriptome. And now when we compare the two lists, we can find the ones that are only belong to this one, not the other one. And then we can define different gene lists based on these properties. And then, surprise, this list have specific functions. The hypomethylated ones were involved in cellular metabolism, and the hypermethylated genes were involved in synapse function. We don't know why. We start to probe the process, so we track each of genes from the transcriptome space to the epitranscriptome space, and we can see indeed this gives us much better resolution of these gene classifications. And that's where we start to see the functional partitioning. It's interesting, a new concept. So we just published this last year using a Japanese bento box as to illustrate this concept. So this is a bento box in Kyushu Shinkansen. And as you see, the Japanese bento box use extensive partitioning to keep the food separated and fresh and good. So I think the cells may also use this kind of partitioning mechanisms to put our chemical modifications on very specific, RA molecules. And this illustration is generated by your friend. Yeah. So you see that the science is done by real people, real person. She's also the first author on this paper. Okay. So obviously this is a mystery why this kind of modifications are associated with intellectual disability in humans, but we are starting to probe the mechanisms, and hopefully that this kind of this effort will give us some clues hopefully this will give us some clues on to design mechanism-based therapeutic approaches for treatment. Okay, so I think that's all I want to say. So today, discussion topic, sort of you can think it's irrelevant, but to me it's very relevant. I have been wondering for some time this question: are we still Homo sapiens? We have been Homo sapiens for 30 million years now. So I want you to discuss that whether we have evolved from Homo sapiens or not. So you have 15, 20 minutes to talk about this. Any questions? Okay. Okay, let's break up to different groups. Like we always do, 4 or 5 people. Okay, shall we start? You ready? I think we should start so we can finish on time. Do you want to start this time, Taiwan?
Sorry, you go first.
All right, thank you. Oh, you said we go first or you go first? Or you?
We start later.
Oh, okay. All right. So okay, we start from this group.
Yeah, we discussed a bit, but according to our knowledge from textbook, the species is defined by the ability to interbreed with each other and exchange different genes. So we think although our brain structure or neural nervous system may get a bit more sophisticated, but we still have the same general anatomy structure and the general gene expression system and protein expression system. So we agree that now we are still Homo sapiens.
Very nice. So I like this answer a lot because you have a clear logic of how to, what is the definition and when we, when we define Homo, the species, what kind of criteria we use this. So I think it's a very good answer. I really like it.
And we firstly we discussed about the definition of Homo sapiens, and according to today's definition of Homo sapiens, we are still Homo sapiens because genetically we are the same for a long time. But after that, we discussed about definition of evolution, and today's definition of evolution, we are still Homo sapiens, but if we think about the new definition of evolution, such as caused by education or some other environmental factors, maybe we are not Homo sapiens now, because for about two we can differentiate it by some education or social environment, and we change here. We have changed for we have changed. mm, so we are we evolved from previous Homo sapiens. So today's definition of according to today's definition of evolution, we are still Homo sapiens, but when considering the new definition of evolution, we may not Homo sapiens. It may not be Homo sapiens.
Sorry, I didn't turn on the microphone. So the second group is saying by the current definition, we're still Homo sapiens. But if the definition, if we have to decide a new definition, like, just now we have new definitions of a gram, a meter, and all those things, then maybe we're not. What do you think that makes us not Homo sapiens anymore? What do you think is the biggest difference?
The difference is in culture or culture.
Difference of biggest.
Yes.
Interesting. Yes, very good answer. Thank you.
Thing, things we can do is increase because of the development of tools, but there has no big breakthrough such as the transition from four-legged walking to two-legged walking. So we think genetic information and phenotypes have not changed. However, we don't know the truth without checking the genetic information of the ancient people and modern people. That's all.
Say the last sentence again. What did you say? What is the last of 1,000 people? What's that?
We think certain genetic information and phenotypes have not changed.
Okay. Okay.
But we don't know the truth without checking the genetic information of the ancient people and modern people.
I
So
see.
we don't know the truth.
So you're saying we only know the reference human genome in 2002. So so now there's some ancient genome hunters, right? So without further information, it's hard to see how much our genome has evolved. So so do you think we are still Homo sapiens or not? It's a binary question. It's not or maybe I should add maybe, or what is your answer? If A, yes, B, no, C, maybe. You can always go to maybe, I guess. What do you think? What is your group's conclusion?
Once more, please. Sorry.
Are we still Homo sapiens? That's my question.
Yes, we still Homo
Yes, we
sapiens.
are.
Yes, yes.
Yes, we are. Okay. Yes, we can. Okay, great. Thank you. Yes, you, right there. You. Yes, you can.
Uh, we think we are Homo sapiens because
Okay.
my father is Homo sapiens, my mother is maybe Homo sapiens,
Maybe.
Homo sapiens.
Okay.
They, I am their child, I am Homo sapiens. で、future, I get married. で、I get baby. I will get baby. Maybe this baby is Homo sapiens.
So where did the first Homo sapiens come from?
My father is Homo sapiens. はじめ起源。Homo sapiens 起源。なんだろう。difficult. いいな。起源。
Okay, but
Oh,
your logic
sorry.
is okay. My dad is Homo sapiens. My mom is Homo sapiens. Therefore,
Oh,
I am Homo
right.
sapiens too. I like the logic. It's fine.
Yes.
You know, it's very clear. You have your logic, and then you give me a clear answer. Yes, we're Homo sapiens. I am Homo sapiens. Great, perfect.
What?
It's fine. Yeah,
Thank you.
yeah. That's what I'm looking at. There's no right answer or wrong answer. But when you answer something, you give the logic. You say, "This is my conclusion, and this is why I think it's this way." But I don't agree with you, but that's okay. That's another question. But the answer is a good answer. Congratulations.
Thank you. Thank you.
Our group think we are still Homo sapiens, and I heard a word called Homo sapiens sapiens. That is a subspecies used to distinguish anatomically and behaviorally modern humans, which we are from those of our archaic human ancestors. And maybe we have some genetic difference from the archaic human ancestors, but I think we still belong Homo sapiens, and we're still using the term Homo sapiens.
All right. Okay. Okay.
Thank you.
Anything from your group to no? But it's not clear to me why. Why you think there's not a huge change, dramatic change yet to define us as a new species? No, you don't think it's happening. Okay. Well, that's a good answer. Thank you. Okay.
99% DNA are shared in Homo sapiens and we us. But Homo sapiens can't use words as well as we use. Homo sapiens and we have differences of the DNA modification. So we are not Homo sapiens.
We're not.
We're not Homo sapiens.
Why? And because we have different DNA modification?
Yes.
But Homo sapiens have DNA modification.
We
And
have,
now,
we have different modification from Homo sapiens.
we have different what?
We have different modification from Homo sapiens.
Really? 99% of the genes are shared between mouse and human. But we're not there is not a human-specific DNA modification yet. Not, you know. So all the molecular DNA modifications that are found in human can also be found in other animal species. So I don't think you can use DNA modification to define the species. I think something else. But that's an interesting approach, I think, to look at DNA chemical modification, right? So that I like that to find the right angle to answer this question. Okay? But that's a good answer. We're not. Your group agreed we're not. Okay, great. How about Taiwan? Thank you.
Thank you.
What's your conclusion?
Hello. We we think we are definitely different from the most original Homo sapiens, but we are still Homo sapiens. Uh, because now we have many technological products can help us to cope with problems, and we don't need to do anything by our own. So maybe we lose some ability of the original humans, but we are still humans. Thank you.
All right. Thank you very much. Yeah, you have something. You want to have something to ask? Oh, I didn't know. I thought that he represent. Yeah, sure about that.
Oh, I'm sorry. So this group, we thought that, well, if the plain question is if you are still the same species as before, if you're still Homo sapiens, the answer will be yes, because the definition is basically that we can interbreed with each other, and this we haven't reached this point yet. However, if the question is if we have evolved from the previous Homo sapiens, then we may say that, yeah, probably yes, because, well, we have changed many, many times our environment, and it is known now that the environment changes our genome, and this genome can change our brain structure and how we act with each other or our characteristics, and it can have a gain of function or loss of function compared to before. So in that sense, we have evolved, but not enough to be a different species. There's their answer. Thank you.
Um, Very interesting answer. And I'm really happy that you know put the sort of message from my class, my own research into this question. The reason I asked you this question is, in your generation, my generation too, that we're experiencing a huge technology advance. I think that is a key, the key issue that I want to mention. For the first time, human, only us, we can do genome editing on ourselves. And that, to me, is a definition line. Like you said, definition can change because we do not need interbreeding to change our genome anymore. We can edit our own genome. And this is a privilege we have only in this generation. Another key technology advance is the internet, right? Everybody, we're all humans. You can call it the cyborg, right? We're in a sense, we're all this human-machine hybrids, like the Terminators, you know. Um, and this is already pretty old. This is in 2015, right? This is also approved by FDA for treating blind people. What you do is you put a little camera, and the signals are sent to the tongue, right? So previously, a machine is only assisting human, but doesn't change human. But with this machine, the tongue has to learn how to interpret the information here, which comes from the camera. So now the machine actually, when you put it, so it really taps into the brain plasticity. So the brain has to learn how this machine works, right? How to interpret the signals from the tongue. And the tongue now is used to sense visual information. So this is a transsensory information. And this is also, if you think that, this is also a way to direct our own evolution. If we think that our eyes are, have this evolution, our tongues are having this kind of evolution, now they're connected. This is transsensory evolution. So, and now there are more and more devices like this. For deaf people, you can put a little vibration device on their backbones, right? And this vibration is detected and transmitted directly into the brain. And again, the brain has to learn how to interpret this information. So this from this generation right now, I think we are living in sort of like a new age that technology can direct our own evolution. And by inventing them, we are really controlling our own genome information, our own, the use of our bodies. So some people, provocative people have started to call us Homo evolutus because the definition changed here, the game changed here, is that we can edit our own genome, and we can control the way that our sensory information or our organ evolution. So again, there's no right answer or correct answer. You just but you can have your own answer. That's all I want to let you know through this class. Okay. So sorry that we're a few minutes late, but does everybody have a good break? There's no break. Lunch break and homework in July. Okay. Okay, Taiwan, nice having you. Okay, class is over. See you next week.
Okay, thank you very much. See you.